This video is going to be looking at the process of a muscle contraction and briefly describing the different types of muscle contraction. So first off, we're just going to start with an overview of a muscle contraction. So we start with an electrical impulse being sent around the body. We then look at how these impulses travel across and through the neuromuscular junction. And then the most detailed section is the sliding filament theory, which is what happens within the myofibril in the muscle. So we're just going to talk about how nerve impulses are transmitted. So we start with a stimulus, which is just a change in the environment of an organism. For example, a ball coming towards a goalkeeper. The goalkeeper is obviously going to want to catch the ball. So the stimulus is detected by a receptor, which can be muscles or glands, but in this example it's the muscles in the eyes that detect the approaching ball. The receptors then send an impulse to the central nervous system via a sensory neuron. This tells the brain that the ball is approaching and what they're going to do about it. The central nervous system processes this information and send a new impulse down a motor neuron to the muscles they want to contract. The effector then receives this impulse and contracts according to what needs to be done. This is where the sliding filament theory happens, but I'll get onto that in just a minute. So the effector brings about the response. In this specific example, the goalkeeper contracts their biceps brachii and brings the ball into their body, catching the ball. So we're now just going to look at the neuromuscular junction. So we're backtracking a little bit to when the central nervous system receives an impulse and then sends a new one down the motor neuron. Once the impulse reaches the end of the axon, which is just the cell membrane that surrounds the long fibre of the neuron, they approach the synapse. So the impulse approaching the synapse triggers the release of a chemical neurotransmitter called ACH, or acetylcholine. So this chemical then diffuses across the synapse and binds to receptor sites on the muscle. So if there is enough ACH that is diffused across the synapse into the receptor sites, then the impulse can continue down the muscle. So just look at the picture there, it's got all the labelled sections. So we've got the vesicles coming to the bottom of the synapse and then them diffusing across the synaptic cleft as it's labelled there. But before we continue with the impulse, we're quickly going to just have a look at what the muscle is like at rest. So the thin myofilaments within the sarcomere of the myofibril are called actin, and they have a binding site. So at rest, this actin binding site is covered by two proteins called tropomyosin and troponin, as you can see in the image. Also, in the image you can see that tropomyosin is a thin, long strand of protein, whereas troponin is globular, a more globular complex. So these two proteins are preventing actin from binding to a thick filament myosin, which is, so to speak, energized with ATP, or as it's properly called, adenosine triphosphate. So it's ready to bind to these active binding sites. Now, in the second image, we can see that the H zones and the I band are at maximum width here. So now we're just going to go back to the impulse. So it goes through the cell membrane of a muscle, which is called the sarcolemma, and then the T-tubules carry the nerve along the muscle fibres. So the impulse then reaches the calcium store, which is called the sarcoplasmic reticulum. This then triggers the release of the calcium ions. The calcium ions arrive at the sarcomere, which is a contracting unit, and then bind to troponin, changing its shape, which also changes the shape of tropomyosin. So as a result of this, they both peel away from the actin binding site, leaving it free to bind. The myosin obviously then attaches to the binding site using the ATP that it's got. So as I've just said, the myosin binds to the actin binding site using the molecule of ATP, forming a cross bridge shown in the image. As you can see, the myosin arm attaches to the actin, forming the cross bridge. So the myosin arm pulls the actin towards the middle of the sarcomere, which is also known as the M lines. This is therefore sliding the actin along the myosin from both sides. So the process of hydrolysis has meant that the ATP has been used and broken down into adenosine diphosphate, which is ADH, and a single phosphate. Therefore, the myosin arm needs more energy, so need a new ATP molecule. Once a new ATP molecule is available, as a result, the myosin releases from the cross bridge and gains the ATP. Once it's energized, the myosin arm will be able to rebind with another actin binding site and repeat the power stroke process. Right, so whilst this is happening, it's occurring at every myosin arm that's in the sarcomere, which is why the muscle shortens. So there's many actin-myosin cross-bridges forming and completing the power stroke. So the overall effect of this is that it makes the sarcomere shorten. So what's happening is the different sections of the sarcomere are overlapping with each other. So the Z lines are moving in closer towards the middle, or the M line. The I band and H zones are getting a lot smaller, but the A bands aren't actually changing length. So now we're just going to look at how the contraction is ended. So a fully contracted muscle, the H zones within it have disappeared completely, and the I band has completely narrowed all the way down. Also, the ends of the actin myofilaments have overlapped, as well as the A band remaining an unchanged length. So once a response has been performed, so for example once the goalie has caught the ball, the central nervous system will stop sending impulses down the motor neuron. The myosin will then release from the cross bridge and gain an ATP molecule, like it has been doing previously. But before it can form another cross bridge, the troponin and tropomyosin cover over the actin binding site again, leaving the myosin energized and ready for the next contraction. Also, the calcium molecules return to the sarcoplasmic reticulum. So finally, I'm just going to go through a few different variations of muscle contraction. So firstly, there is one type of isotonic contraction called concentric, which is when the muscle shortens during a contraction. And the other type of isotonic contraction is called eccentric, which is when a muscle lengthens during a contraction. So an example of a concentric contraction is the biceps brachii during a bicep curl. And an example of an eccentric contraction is the biceps brachii when the elbow is being extended back down from bicep curl. So another type of muscle contraction is called isometric, which is when the muscle length stays the same during a contraction. So for example, in a rugby scrum when you're pushing it against opponents. Now the final type is called isokinetic, which is when the muscle contracts and shortens at the same speed. So these contractions allow muscles to gain strength easily and evenly throughout all the range of movements. So these are why they're often very useful when improving muscular strength after injury.